Recall, the kidney maintains acid-base homeostasis through three mechanisms: bicarbonate reabsorption or reclamation at the proximal convoluted tubule, bicarbonate generation at the collecting duct, and hydrogen secretion, buffering, and excretion. Now let's look at the second way the kidneys maintain acid-base homeostasis: through bicarbonate generation. The proximal convoluted tubule reclaims the majority of the filtered bicarbonate. However, bicarbonate generation occurs at the cortical collecting duct. Not only must we reclaim our bicarbonate, we must regenerate the bicarbonate used up, buffering our acid load. As you recall, we need to produce bicarbonate to balance our daily acid load from protein in our diets. Proteins are metabolized into amino acids and eventually hydrogen ions. As discussed, bicarbonate generation occurs at the cortical collecting duct. Remember, there are two important cell types here: the principal cell and the intercalated cell. In general, you can see the mechanisms involved in this process. Importantly, in order to make new bicarbonate, the kidney must secrete a hydrogen ion. Let's discuss in detail how this occurs. Bicarbonate synthesis occurs when CO2 from the blood combines with intracellular water to form carbonic acid. This is again with the aid of the catalyst carbonic anhydrase. The carbonic acid goes on to dissociate rapidly into a hydrogen ion and a bicarbonate anion. This time the hydrogen ion must be secreted and excreted so as to not contribute to the overall acid load. The bicarbonate is then reabsorbed across the basolateral membrane in exchange with a chloride anion using the bicarbonate chloride exchange transporter. This exchange maintains electroneutrality. The entire process results in a net secretion of one hydrogen ion per one new bicarbonate for reabsorption. For the cortical collecting duct to successfully secrete a hydrogen ion, the following must occur. 1. Passive sodium reabsorption down its gradient through the apical epithelial sodium channel, ENaC, found in the principal cell, thereby causing a negative charge in the tubular lumen. This sodium gradient is maintained by a sodium-potassium ATPase. 2. A negative charge in the lumen promotes the movement of other cations like potassium and hydrogen into the lumen, thereby maintaining electroneutrality. Finally, 3. Hydrogen secretion is facilitated by the active transport via hydrogen ATPase located in the apical membrane of the intercalated cell, whereas potassium moves passively across the apical ROM-K channel found in the principal cell. However, we're not done yet. The hydrogen that was just secreted at the cortical collecting duct must now be successfully excreted. The cells within the distal cortical collecting duct are impermeable to hydrogen, thus preventing reabsorption. High hydrogen content in the urine can be very acidic and corrosive. While some free hydrogen ions are excreted in the urine, the majority must be bound by buffers, and reduced buffering capability in the urine impairs hydrogen ion secretion and excretion. The two major buffers in the urine are ammonia and ammonium trapping, and titratable acids. Titratable acids are anions that are filtered through the glomerulus and travel distally to bind with hydrogen at the cortical collecting duct. They mostly come in the form of phosphates, but can also be sulfate, hippurate, and citrate. The amount of titratable acids that are filtered are constant, meaning they cannot be increased or decreased based on dietary intake and acid load. Ammonium trapping is the major urinary buffering mechanism. At the proximal tubule, glutamine is metabolized into glutamate and free ammonia, NH3. Ammonia is lipid-soluble and diffuses freely into the tubular lumen. Ammonia has a high affinity for hydrogen in the tubule, buffering it to become ammonium, NH4+. Since ammonium is impermeable to the apical membrane, it is trapped in the urine for excretion. The production of ammonia varies with protein intake and daily acid load. In fact, chronic metabolic acidosis results in reduced intracellular pH, thereby stimulating glutaminase. Glutaminase is the enzyme responsible for converting glutamine to ammonia, increasing its buffering capacity. The opposite is true, whereby high intracellular pH, alkalosis, will suppress this mechanism. Interestingly, low intracellular potassium concentrations stimulate glutaminase activity. Likewise, high intracellular potassium concentrations will inhibit glutaminase. These mechanisms are relevant when we discuss the relationship of potassium and acid-base status and renal tubular acidosis disease mechanisms. To summarize, the excretion of a hydrogen ion and generation of bicarbonate at the cortical collecting duct requires the following mechanisms to be intact. 1. Sodium delivery to the cortical collecting duct. 2. Low intracellular sodium concentration generated by the sodium-potassium ATPase. 3. Sodium reabsorption by the ENaC at the apical membrane, generating a negative intraluminal gradient. 4. Hydrogen ATPase activity and hydrogen excretion. 5. Impermeability of hydrogen efflux back into the cell or extracellular space. 6. The generation of ammonia, NH3, at the proximal convoluted tubule and ammonium trapping. Each of these steps can be physiologically modified or impaired, impacting the kidney's acid-base handling. Breakdown in any one of these processes may result in a non-anion gap metabolic acidosis, also known as anagma, and more specifically, a distal renal tubular acidosis, or RTAs. An important point to make is that the cortical collecting duct is also capable of bicarbonate reabsorption. A general rule of thumb to keep in mind is that no new bicarbonate synthesis can occur until bicarbonate reabsorption is complete. In short, this means if bicarbonate remains in the tubule and is delivered to the cortical collecting duct, it will be reabsorbed before it generates new bicarbonate. That's right, the cortical collecting duct can and will reabsorb bicarbonate first. In fact, under normal conditions, meaning a non-acidotic state, the filtered load of bicarbonate is so large that the cortical collecting duct does quite a lot of reabsorption, and not until it reaches the most distal portion of the cortical collecting duct will there be any new bicarbonate generation. This is done to tightly regulate bicarbonate levels in the body. The kidney must reabsorb bicarbonate and generate bicarbonate when receiving a high acid load. However, under normal physiologic circumstances, the goal is to maintain normal physiologic levels of bicarbonate in the blood. As soon as we exceed normal physiologic serum bicarbonate concentrations, the kidney downregulates reabsorption, thereby increasing bicarbonate excretion, as demonstrated in this graph. Now, let's briefly discuss how and why this occurs. In a normal physiologic state, ammonia production is downregulated. Now, instead, bicarbonate serves as the buffer delivered to the cortical collecting duct. Hydrogen secreted by the hydrogen ATPase and bicarbonate will form carbonic acid and rapidly dissociate into carbon dioxide and water. The water is excreted, but the carbon dioxide freely passes across the apical membrane into the cell. Carbon dioxide then combines with water in the cell with the aid of carbonic anhydrase, forming carbonic acid, which then dissociates into hydrogen ion and bicarbonate ion. The bicarbonate is then reabsorbed in exchange for chloride. Notice how in this setting the hydrogen ion is recycled instead of excreted. Now let's cover a clinical correlation. As we briefly discussed previously, severe or chronic diarrhea can result in a high volume loss of bicarbonate, resulting in a non-anion gap metabolic acidosis. So in this setting, how do you expect the kidney to respond? If you answered by increasing hydrogen ion secretion, enabling bicarbonate generation, as well as upregulating ammonium excretion, you are correct. Now the next question is, what would you expect the urine pH to be? Correct also, it should be low, less than 5.5 approximately. Now let's briefly explore what happens when hydrogen ion secretion is impaired. These types of disorders are referred to as distal renal tubular acidoses, or RTAs. There are two types, type 1 and type 4. Both impair hydrogen ion secretion, resulting in a non-anion gap metabolic acidosis. Additionally, due to the impaired ability to secrete hydrogen ions, the urine cannot be acidified distally. Ultimately, even in the setting of a metabolic acidosis, the urinary pH will be above 5.5. Contrast this with a proximal RTA, whereby bicarbonate absorption is impaired, but distal hydrogen excretion remains intact. That means in a proximal RTA, the urinary pH can still be below 5.5. In summary, bicarbonate generation occurs at the cortical collecting duct as depicted by the mechanisms here. But don't forget its ability to reabsorb bicarbonate as well. The ability to perform this activity is an important mechanism that differentiates urinary pH measurements in proximal and distal RTAs. We'll discuss this further in the future.